So this talk about tarbot involvement in the AQUA-FAANG project. I have two parts. In the first, I will make first a short presentation of the interest of the tarbot and the flatfish in the different work packages of the AQUA-FAANG project, and the results we have obtained until now. And in the most important part, Oscar will present the results related with the analysis of the attack chip and RNA-seq data in turbot immunomaps. So as a short introduction, I will take you to the world of the flatfishes that here I have emphasized the two main importance, both related with the evolutionary adaptations of this significant fish group, and also the relevance for the aquaculture as a very delicious meal and a high commercial value. So regarding the breeding programs in turbot, the selection, genetic selection programs started 25 years ago. Currently in the world, there are being produced 7,000 tons. Despite the production not too large, the high commercial value estimates or stimulates the investment by the turbot companies. The main targets for the turbot aquaculture are the growth, of course, as in most species, diseases, and also obtaining all female populations, because the bigger size of the females in this species regarding the males. But other new flatfish species have been introduced or included in the AQUA-FAANG project, because in the work package 6, we have a particular activity related to the comparative genomics of the flatfishes, and we have analyzed the genome, as we will see later, of the Senegalese sole. They currently only 3,000 tons, but it's an emerging species with increasing importance. So in 10 years, the sector is thinking about 30,000 tons for the 30s. Regarding the Senegalese sole, again, the growth, the sex determination, because the females grows quicker than the males, but also the reproduction are the main targets. Flatfishes from the developmental point of view, so related to the maps, are very interesting because they suffer in a very short period of 5–10 days sharp reorganization of the body. And the work package 1 in this species, we have analyzing only the regulatory elements before the hatching. But in complementary projects, we are interested in analyzing also the main pathways involved in the metamorphosis of this species. Further, regarding the evolutionary importance, the flatfishes acquire a very important adaptation to live in the sea bottom, and so there were a quick evolutionary radiation 80 million years ago that provoked a very bitter controversy about the origin of the flatfishes. Finally, it has been demonstrated that the flatfish morphology was acquired 2 times at least along the evolutionary processes. And also because this quick adaptation and the revolutionary radiation, the molecular evolutionary rate is much higher in the flatfishes than in the sister orders of the perciforms. Good for us, the flatfishes genomes are very compact, so between 500 and 700 megabases. So the amount of repetitive elements is between 5 and 8%. So we have got very consistent and robust and contiguous genome assemblies. First, within this project, we assembled in collaboration with the Norway and Edinburgh groups the genome of the turbot and identify the sex-determining gene as an important tool for obtaining all female populations. But recently, we have also assembled the genome of the Solea senegalensis, the Senegalese soul, and also identify the sex-determining gene with the same aim as in turbot. During this project, Oscar, within an activity related with Work Package 5, integrated all previous information from the genomics point of view, both association studies and functional genomics studies that was taking place since 2007 until now. And he could identify target genes related with specific pathologies, but also other transversal genes that were responded to any pathology related with bacterial, virus, or parasites. So finally, within the project, we have constructed all the libraries, all the expected libraries with very few replicates failing in some tissues in body maps. But in immunomaps, Oscar has finished all the analysis for the attack chip and RNA-seq, and he will present the information on the paper he's writing right now, in the pipelines he has used, and he will post the important doubts that still remain on this point. And the maps has finished the attack running, the pipeline still waiting for the chip, and the body maps is running the attack pipeline and waiting for chip. So that's all by my side. Oscar, you have the floor.
Okay, thank you, Paulino. Hello, everyone. Just let me very quickly share my screen. Oh, I think that Paulino, you need to stop sharing the screen.
I'm trying, but
okay, no problem.
I don't know why this is. Okay. Okay, now.
Yep, now it's on. So, can you hear me well, or is there any problem with the audio?
Yeah, we can still not see your slides, Oscar. I don't know if I am sharing still.
Yeah, we can't see it here either.
Yeah, but now, okay.
And now you can. Yeah. So let me very quickly. Okay. Well, so hello everyone. So what I'm going to share with you right now is the work that we are conducting on analyzing the functional reglomic data on the immunomaps for the AQUA-FAANG project related to turbot. So as you can see here, well, I don't know if you can see the pointer. Let me quickly change to the laser pointer. Yeah. So as you can see here, this is still work in progress. This is our first time tackling the analysis of ATAC and chip-seq data. So many of the things that you are seeing here are can possibly change. But what you can see here is the experimental design of the. Sorry, there is a problem with the. Okay, yeah. So what you can see here is the experimental design of the immunomaps. It consisted on two different kind of immune stimulations, one in vivo in fish, and sorry, I think there is some microphone.
No hay manera. ¿Dónde están las llaves de tu hermano?
Yep. One sec. I think I can silence.
I think that Belén has opened the.
And now I think it's on. Yeah. Okay. So yeah, here is the experimental design for the immunomaps. It consisted on two kinds of immune stimulation: one in vivo in live fish, and another one in vitro using primary cultures of leukocytes isolated from the head kidney of turbot. And in both cases, we used two mimics for a mimic for viral infection in the form of poly-IC and another one as a mimic of bacterial infection, which was the Vibrio. And what we wanted to see here was how each one of these immune stimulations resulted in difference in changes on the expression and the conformation of chromatin at the stage of early infection. So mainly focused on early immune response. And I can here. So this was a multidisciplinary experiment. So it took part in many different universities. So the standardized protocols were carried out by the Wageningen University and the Aberdeen University. The protocols for ATAC and ChIP-seq library preparations were developed by our colleagues at Norway in NMBU and the people at Rosling. Those protocols were then translated by us to Charcot, and now the bioinformatic work is being done by us, but with the help of the people at Rosling from the McQueen's team that have helped us really kickstart the analysis of ATAC-seq and RNA-seq. So here I provide you a few of the resources that we were using to dive into this task. So this first article that I'm sharing here is almost a one-to-one translation of the work that we are doing in Immunomaps, and that has been done in alveolar macrophages in pig. I believe this work is from the switching project, I think. Besides that, those are very well known from in the AQUA-FAANG project, the Hitchhiker's Guide to ATAC-seq data analysis, and this one that lets you explore how ChIP-seq analysis can be done. And then we have the course that was carried out by Embel 2 years ago. I think most of the classes are still uploaded in the AQUA-FAANG YouTube channel, I believe. And then this small course on introduction to ChIP-seq that was done by HBC. And besides this data, here I have the core pipelines that we use to analyze this data. This was the NF core pipelines that take care of the main stages on the processing of data from the sequencing raw data to the post-processing. And here I think I can start now looking at RNA-seq, that is very quick to see. So here I have the PCA plots for the in vivo and the in vitro analysis for each of the conditions that we have tested. And what you can see here is what we will see in also the ATAC and the ChIP-seq, that you can see some clusterization, but it is very widespread, especially in the in vivo. It's difficult to see that clusterization of the samples. In the case of the in vitro, there is a pickling or a big separation of the Vibrio samples in respect to the control and the poly-IC immunostimulated samples. But this is what we got. So before running the differential expression analysis, we did a small filtering. Sorry, can you see all the presentation? Or, okay, right now, yeah. So what we did before running the differential expression analysis was 2 filtering steps. One, making sure that all the transcripts that had a TPM above 5 were retained, and also keep only the transcripts that were represented in at least 2 replicates in one of the conditions. With that, we got a collective transcriptome for the Immunomaps that was around 12K genes. And we ran the DSEQ analysis, and this is the results in a graphic format and in a table format. So here, well, what I'm showing here are all the significant differentially expressed genes across each of the conditions. This was done testing each of the mimics against each of the infections with the mimics against the controls. So here we have the complete list of significant differentially expressed genes, here with a fold change of above 1 in absolute values and so on. And here we have a preliminary analysis on the significantly enriched GO terms that we are getting for each of the analysis. So most of them make sense. Most of the downregulated pathways are focused on cell cycle metabolism and in the differentiation of other blood cells that are not leukocytes. And in the case of the upregulated genes, in general, it's a very big enrichment in pathways related to immune and defense response, overall activation of the transcription and the traduction. In the case of the poly-IC infections, you can see that there is also big enrichment in pathways related to the IFN. And also we got this particular case with the samples in the in vivo vibrio, where we got some functions related to syncytium formation and cell-to-cell fashion that we are not really sure why they are showing there. From what I researched, in most cases, these pathways are shown in infection by some virus, some kinds of in cells infected with some herpes virus. You can see a certain enrichment in this function, but it's not found in any bacterial infection. So maybe if I don't know who is here right now in the course, but maybe we can chat a little bit later with other people involved in other immunomaps to see what they were getting. And then, well, this was the analysis for each of the categories. We also conducted search for common and specific genes in each of the comparisons possible. We need still to dive into each of the gene lists individually to look for specific genes that we are interested in. But here you have also the significantly enriched GO terms for the common upregulated here, and the common downregulated for the in vitro experiment. And here some of the enrichment for the up and down regulated for each of the infections specifically. The ones that are not shown here is that they didn't show any significant enrichment for any of the body functions. And the same goes for the in vivo, not much enrichment here. And also here we can see again this particular enrichment in syncytium and myoblast formation that it's very conflictive to what we know about viruses. So that's it for RNA-seq for now. Now looking again at the ATAC-seq data, this is the output of the running NF-core library, and as you can see, it's the same situation. Very little clustering for each of the conditions, with a slight deviation of vibrio in comparison to the controls and the poly-IC immune stimulations. And this is the case for ChIP-seq, with some really weird cases, particularly here in the in vitro. In the case of in vitro, for the immunoprecipitation of the K27 trimethylation mark, that it's related with inhibition of genomic regions. So now after running FCOR for the ATAC-seq and the ChIP-seq, one of the first things that we did was construct a blacklist for the ChIP-seq regions. This is especially useful to avoid genomic regions that frequently produce artifacts and noise, particularly in ChIP-seq analysis. This is meant to improve the signal-to-noise ratio on the peaks that are found in the good regions that are not found in the blacklist. For this, we are using the controls of the ChIP-seq experiments that we are calling the inputs. And how we did that was using two programs, UMAP and Blacklist. This is very well explained by one of our colleagues in the Roslin Institute, it's Paulino. So what we are basically doing is with UMAP, we quantify the mappability of a genome for a given read length of size that we define as K. It can be K100, K150, or so on. And it generates all possible k-mers of the genome that match this size. So then it maps the unique k-mers to the genome using bowtie, I believe, and it marks the position of each k-mer that aligns to only one unique region. After that, these mappability files that generates UMAP are passed to the other program, to Blacklist, and along with the BAM files for each of these inputs, we can generate the Blacklist. So for this kind of procedure to generate a Blacklist, it's good to put as many inputs as you can. As far as I know, the development of a Blacklist is not experiment specific. So as you can see here, even though I'm talking about the immunomaps, we are using inputs from the body maps, the maps, and immunomaps. And if we have inputs for chip experiments on from other projects also in Turbot, we would have added those. Those will help with the specificity of this Blacklist. Here is the output of this pipeline. Basically, around 7% of the Turbot genome is collected in the Blacklist. Here you have the representation for each of the chromosome. And here we can see that most of these blacklisted regions are found near the telomeric regions, near the centromeric regions, and in some instances they are found along other positions in the chromosome. So besides the blacklist, we wanted also to filter a little bit those the replicates that we were getting here. One of the things that we did was visualize the big files that are generated by NFCore using simply IGP, and we were looking to find stuff like this, those serrated and jagged profiles. Those are bad news. This usually means that for those samples we are getting very small peaks and very small representation of the overall genome. So we excluded that, and we also looked at the peak counts that we were getting also from NFCore and excluded those that were below a certain threshold. So in this particular case, we were excluding this one, which corresponds to this serrated profile, is the replicate 2, and also we excluded replicate 3. We ended up including both these two. So the final step that we did for this data curation was running IDR to get the highly reproducible peaks. This is a very complex system, so I will not be explaining it here because I will probably butcher the explanation for it. But basically what it does is for each of the conditions, you compare each replicate pairwise with another replicate, and then you establish a consensus list of what are the highly reproducible peaks that we are finding here for our datasets. So after running all of this, one of the things that you can do is start with ChromeHM that was previously talked about yesterday and also today. So basically, ChromeHM, what it does is it's a software that conducts the learning and characterizing of the chromatin states, integrating many different datasets. In our case, it was data from ATAC-seq and ChIP-seq for 3 marks. And it models the presence and absence of each mark using a hidden Markov model, which is given by this HMM of the name. So usually what you will find if you search in Google other papers that use this program, usually for 3 marks class tag or for 4 marks, they usually go with 10 chromatin states, 12 chromatin states. Those are given by the researcher to ChromHM to establish how many chromatin states you want to force the model to fit. So basically, what I would recommend, based on what I learned from the people at Rosling, is in this particular case, iterate from 10 to 15 states and see what you get. This is the result that we are getting for the in vivo stimulation and the in vitro. As you can see, in the case of the in vitro stimulation, we ended up with less than 10 states. That's because the other 2 states that we were getting didn't really, we didn't got any real explanation or biological explanation on how those were generated. So we ended up reducing a little bit the number of chromatin states. And these graphs are the output of ChromHM. This one is the job of the researcher to do. But right now, between studies on human, on traditional cattle, other model animals, it's fairly easy to establish this chromatin state. And this is a expanded view on what you are getting from ChromHM. So basically, the emission parameters for each of the marks, as we have seen before, the state enrichment for different genomic features. And this that is particularly useful to identify the promoter regions on the active TSS and so on, which are given by this TSS, that is transcript start site. And as you can see, for the chromatin state 5, 8, 9, and some in 10, those are related to TSS regions. So next step was for us using DeepBind. DeepBind works fairly similarly to DSEC2. In fact, I believe that it uses DSEC2 to carry out some of the steps. You can run it in just one command, but this is the extended the extended version of using DeepBind. Basically, you just read the BAM files and the bed files that you want to provide. You count the peaks and generate a consensus list for each of the conditions. Then carry out normalization. You define the groups that you want to compare. In this case, we wanted to compare each of the mimics against the controls, and then do the analysis. So this is an important note that we have found, that for running DeepBind, you need to use the complete dataset, presumably, of peaks that you are getting, and not only the highly reproducible or highly confident ones that we were getting with IDR. And this is the results in our case. So in general, the poly-IC immunostimulation doesn't seem to have much effect on chromatin structure. Not so much with Vibrio. In Vibrio, we're seeing a large amount of peaks for especially ATAC and the promoter-associated mark in K4 trimethylation. But yeah, this is more or less what we are getting for each of the marks studied. And going a little bit further on this note, so basically we were running DeepBind using only the complete dataset, not the highly reproducible ones. But this honestly doesn't make much sense to us. I don't know if someone can point us in any direction, because in the case of DSEC2, usually you can run it on filtered sets of transcripts. So we don't really know what is the reason that we can't use DeepBind to run our highly reproducible peaks, but because as you can see, we are getting very different outputs from one to another, and we don't know if this running the highly reproducible peak only dataset is just noise and artifacts, or if it's truly meaningful, because If it's not, it will really improve our output for K27AC and K27ME3, and also with poly-AC infection. So that's it for this sec. Then once you have all the peaks, the list of peaks that you want to analyze, mainly in our case were the highly reproducible ones and the differential sets of peaks, we run the annotation using this software, Homer, that is fairly easy to use. This is an example of the output that you're getting for each of the peaks with a simple annotation here on each of the peaks. As you can see, the promoters are fairly easy to identify. They are labeled as so, and how Homer carries out this by looking at peaks that are found more or less near in this window of -1 kilobases plus 100 base pairs. And this is what it considers to be a promoter, but we have seen other works where the promoter region is considered to be a little bit bigger, 2 kilobases, 3 kilobases. The most we found was 5. And for the case of enhancers, these are fairly complicated. So we know that enhancers should be found in intergenic regions and intron regions, but it's difficult to identify with whether those peaks are correspond to enhancer regions or no. And also it's difficult to integrate those enhancers with the genes that they are affecting. We have found that, so some other works, what they are doing was look for the nearest gene, or in other cases the nearest active gene, but we are not very sure what is the best way to accomplish the linking of the enhancers to the genes. The promoters are fairly easy. And here you have the summary for the highly reproducible peaks for each of the datasets. So basically, this is the in vitro attack, in vivo attack, and so on for the marks. K27ME3 in most of the peaks for K27ME3 are found in intergenic and intronic regions. ATAC, K4ME3, and K27AC are fairly similar, but K4ME3, that it's usually associated with promoters, is found more prominently into those, K27 in introns, and so on. This is what we got. And here we have one way that we have found that we can link the promoter regions that we have found in those differentially binded datasets, and how to link those with the differentially expressed genes. So basically here I have information for two of the marks and ATAC. Here are the differentially histone modified regions separated between down and up regulated. Those are the same, but only the promoter regions. And here we have the differentially expressed genes separated between down regulated for each of the conditions and up regulated. We have each of the sets here, and here the amount of differentially expressed genes that are have also their promoter included in one of those differentially histone modified regions. To test this correspondence, we carried out a hypergeometric test. As you can see, especially for the upregulation, we have a big significance for most of the cases where we could establish this comparison. And this is my final presentation. So I have started to look at the motifs that we are finding in the different lists. For this, we are also using Homer with this function, find motifs genome. And what we're getting, among other many files, is this kind of tables. So this one is for the known motifs that Homer has identified for one of our datasets. So as you can see, Homer conducts the analysis in two different ways. On the one hand, it looks for motifs that are known and are recorded in the database. And from what I have seen, it's almost 1K motifs in total. But it also carries out a de novo motif finding in other datasets. This is only the known motifs. As you can see, we have this kind of plots that were presented before by Malcolm. And here we have the name of the desired transcript, the family it corresponds to, some additional information, and we have the enrichment for each of the significance, sorry, for each of the motifs. So we have done that with the differentially binded dataset, as you can see here. And we have established two different cutoffs. So in some works, what they did was only keep the motifs that had a significance about below 0.05. But Homer recommends only trusting those that are found in that have very significant enrichment. So those different datasets. And this was done last Friday, so I didn't have much time to look at those at the list. So I just look at the most prominent transcription factor families that were found. And here I have some of them outlined in black, and here are some of the functions that we have found. So mainly for the most of the poly-IC challenges, we found many transcription factors that are related with the IFM regulatory factors. We also have very strong representation of the BCIP and the ETS families that, among many other functions, include difference and stress response in the case of BCIP and cell cycle differentiation, migration, and apoptosis in the case of ETS. And then some signal for BHLH and homeobox that are related with hematopoiesis and angiogenesis. And this was very weird. So that what this tells us is that we can't really trust the findings of the known motifs, because this mouthful of a transcription factor family is only found in each plant specific. And it's, of course, it's related to with difference, but it's only found in plants. So from what I can see, it's that you really need to be careful on what motifs you are finding for this analysis. This is it. This is my final slide. So we don't have really any concrete results yet. We have generated the bulk of the data, but we still need to dive into it fairly more carefully. So now we think that we can start with the questions. And of course, if you have any recommendation, we are not experts in this, so we are very pleased to hear from you. Thank you.